If you can hear me, please inform me to start. Okay, very good. In the name of what? So today I'm happy that after a long time, more than 1 month, again we can be with each other in this class and we can continue this course. So if you remember, at the start of this semester, we went through the infection and disease, and we discussed about the relationship between host and microbe. And then we explained something regarding what is infection, what is disease, what is infectious disease, and what are different types of infections, pathogenicity, virulence, and which microbes are normal flora and which microbes are pathogens. Then we explained that normal flora come to our body after birthday. From the first minutes and first hours, different microbes come to our skin, come to our intestine, to our nose, and they will be permanent resident there. So normal flora are permanent residents, but pathogens are not resident. They come for a short time to our body and our body defend itself. So in the slide number 10, we discussed about what are the true pathogens and what are the opportunistic pathogens. So true pathogens capable of causing disease in healthy person. If a true pathogen come to our body, we go through disease. But opportunistic pathogens cause disease when host defenses are compromised. Lies mostly regarding about Candida albicans, which is a yeast or mold, Pseudomonas, and some amoeba, some bacteria. They are opportunistic pathogen. So only we get disease when our immune system has problem. Pathogenicity. So when one pathogen come to our body, it has pathogenicity, and the power of the pathogen we call as the virulence. Virulence show how much is the power of the pathogen, and virulence factors are different things like adhesion factors, biofilm, extracellular enzymes, toxins, antiphagocytic factors. So they are different tools. They are different things which microbes, by help of them, can make themselves more powerful. And I think this is, as you wrote, it is the last page which we discussed about. In order to establish a disease, we have 3 things. So first is the port of entry. Some microbes come to our body according to food, according to coming to our mouth, and then going to the intestine. Some come to our body according injury to our skin. Some coming to our body according our nose, according inhalation, coming to our lung. Some coming to our body according injury of our skin. For example, after an accident, there is injury in our skin or burning in the fire. So number one is the port of entry. So if one microbe find the port of entry, then come to contact to our body, and then invasion starts. Invasion to the target tissue. For example, some microbes target tissue is liver, some microbes target tissue is lung, some the others are nervous system. So each microbe, according its metabolism, according which enzymes contain, find the best place for invasion, for attack. So different microbes have different part of body for attack. And in this situation, Haas defenses barrier is working and try to stop it. So there are two things: microbial attack and immune system resistance. If the microbe is more powerful, this is start. If the immune system is more powerful, microbe is stop. Okay? If microbe can in our body continue multiplication and residence, for example, in the blood, after bacteremia, if one microbe like Pseudomonas can continue its residence in blood, then going to the septicemia. So a large number of microbes present in the blood, and special care needed to be done by physicians, like special antibiotics, and trying to stop them. If cannot stop it, the person can be killed by this microbe. If microbe can distribute in the body, can go out from the body to attack the other hosts, the other people. So portal of exit. For example, if we have a viral infection in our intestine, or a bacterial infection, so those bacteria or viruses can go out from the body, from feces or from stool, from intestine, and then make the other person infected. So different parts are possible for coronavirus. For example, attack to nose, to the pharynx, to the lung, and the port of exit is again inhalation, and according cough and sneeze, and can going to the other people and infect them. So as we have different port of entry, mostly the same ports are available for exit. Port of entry, port of exit. So port of entry, attaching, surviving host defenses. If microbe can survive host defenses, so disease with signs and symptoms are started. So now it is the page as you wrote. We should explain with more details. Page number 13. Ports of entry. So when there is an infectious disease, for example, we see one person has COVID-19. So we use mask. Why do we use mask? Because we know the port of entry is our nose. By our nose, this virus can come to our nose, our pharynx, our trachea, our lung, and makes the person infected. Yes. Or when one person has injury, accident, you clean, for example, injured hand, you clean it, wash it, disinfect it, and then cover it with a clean material, not allowing microbes come to the injury. So characteristic route, a microbe follows to enter the tissues of the body. One port of entry is a skin. So any accident, anything come to our skin is look like a barrier. Our skin not allowing microbes going to the live tissue. So any nicks or abrasions or punctures or incisions, anything happening to our skin allow microbes penetrate to our body. Don't forget that those microbes which are living on our skin as normal flora, when they are on our skin, they are normal flora. If by any accident, any nicks, any puncture to our skin, normal flora can penetrate to the inner layers, to the inner tissues, then they are not normal flora. So our muscles, our liver, our kidney, our brain, all of these parts are sterile. There are absence of any microbe. If any true pathogen or opportunistic pathogen or commensals can go inside to the sterile parts, then they are pathogen. They are pathogen. Or for example, in our intestines are full of normal flora. So we don't have any problem because they are normal flora. We don't go to the physician because we don't have any problem. But for example, if any accident happening to intestine and blood coming to intestine, or one person with cancer in intestine or any problem in intestine, if normal flora can penetrate to the blood, to the veins, to the abdominal cavity, so from this time, they are not normal flora. If they can reach to the parts which they should be sterile, from this time, we call them pathogen. So, except on our intestine, in upper respiratory tract, and inside our intestine, the other parts are sterile. Any microbe reach this parts, this microbe is a pathogen. Okay? For example, you want to get a flower, a pin of flower going in your finger. With this, any microbe penetrate the tissue and going to blood without thinking its name, they are pathogens. So they go to the blood, it is bacteremia. So immune system suppress, kill them. So it's okay if immune system cannot suppress and kill the bacterium in the blood, so it turns to the multiplication of this bacterium, and we call it septicemia, and it is very dangerous. Okay. So if you watch or if one child walk on the ground without shoes, so with very small injury, some viruses like warts can penetrate to the skin, to the inner parts of the skin, and make a disease. The other part of the body is gastrointestinal tract. So different microbes daily with food, with drink, and other ingested materials can come inside our intestine. So some of them are normal flora without any problem, and some of them are pathogen. If their numbers are low, normal flora and immune system suppress them. But if we eat food full of microbe, more than what it is infectious dose, so disease will be started. So accordingly, when you prepare food at home, you go through disease less than you buy food outside home, because at home you do most hygiene policies. But outside home, in restaurants, in different places, sometimes hygiene is not used properly, and there are many pathogens in food, drink, or other ingested material, and make us problem going through the disease. Mucous membrane. So in our nose, in our lips, in intestinal layers, there are mucous membrane in the respiratory tract, which it is a part for defensive against microbes. Yes, of course. Urogenital tract is another part. So whole animals, not only animals, as well as human, urogenital tract is sterile. It is sterile. It is absence of any microbe. And from the time urine going out from the body, it is started to be contaminated with different microbes. Okay? Accordingly, if there is any microbe or any microbes in urogenital tract, the person is a patient, and during urination, during the life, has many problems, and going to the physician, and by urine culture, physician can understand it and by antibiogram give appropriate antibiotic and it is stopped. So urogenital tract never should contain any microbe. Urogenital tract doesn't contain any normal flora inside, okay? But very close to the place for urination, maybe a little microbe are present, and outside the urogenital tract, it has normal flora, but inside, nothing. And sometimes some microbes can transfer different microbes, like some protozoa, Trichomonas vaginalis, for example. It is Trichomonas vaginalis. It is a protozoa, or some bacteria, some viruses can be transferred sexually from one person to another person. And the last part is transplacental. Some microbes can transfer from mother to the fetus in the uterus. So some viruses, some bacteria, some protozoa can transfer. If the mother is infected, the infection through the blood, according placenta, can be transferred from mother to the fetus in the uterus. So there are different parts, like broken skin, like by insect bite, like according ear, conjunctiva of eye, nose, mouth, placenta, vagina, and urethra. Different parts are present in our body, which they are used as the portals of entry by many different microbes. Another thing we want to discuss a few minutes is infectious dose, or ID. Infectious dose. What is this? Minimum number of the microbes required for infection to proceed. So, for starting any disease, at least a special number of a microbe is needed. Okay? And it is not the same for different microbes. Some microbes with a few microbes, some microbes with higher microbe, and some microbes with a large number of microbes can start disease. Microbes with small infectious doses have greater virulence. So as much as they have more power, the number needed is less. As much as a pathogen is weaker, the number needed is more. Okay? For example, there is one disease with the name of Q fever. It is done by rickettsia. So only one rickettsia is sufficient for start of Q fever. But regarding tuberculosis, tubercle bacilli, Mycobacterium tuberculosis, or giardiasis, it is an protozoa, it is 10 microbe is sufficient. But regarding to the Vibrio cholera, it is another infectious disease. 10 in power of 9 bacteria is needed. So 1 bacteria is needed over here, but here 10 in power of 9 bacteria is needed regarding to the cholera. So if we drink a glass or some glasses of water and it contains more than 10 in power of 9 bacteria, we get the cholera disease. If the number of the bacteria is less than this amount, we can suffer it, and our immune system can suppress the present bacteria. Yes, we have different diseases like U fever. It is done by Coxiella bureti, tularemia by Francisella tularensis, brucellosis. It is done by Brucella abortus, viral encephalitis by Herpes simplex virus, plago by Yersinia pestis, gonorrhea, which it is sexually transmitted disease from one person to another person. It is done by Neisseria gonorrhea. Anthrax, it is done by Bacillus anthracis. Typhoid by Salmonella typhi, cholera by Vibrio cholera. So here you can see this table. Estimated infectious dose of selected pathogens. So regarding measles, which come to our body according respiratory system, only 1 virus is sufficient for disease started. Q fever, 1 to 10 regarding respiratory. Tularemia, 10 to 50. Smallpox, 10 to 100. Brucellosis, 10 to 100. Viral encephalitis, 10 to 100. Plago, 100 to 500. Gonorrhea, 1,000. Anthrax, 8,000 to 50,000 spores. Typhoid, 10,000. Chlorella, it's needed 100,000 cell ingested for disease starting. So maybe during life, many times we drink food or water containing chlorella, but we don't get disease because their numbers are less than what they can start disease. So in this page, we understand that for starting any disease, microbes, pathogens, opportunistic pathogens can reach to their infectious dose. And different microbes have different infectious dose. You can go through the internet and any microbe, after writing any microbe name, writing infectious dose. And then you understand what is the number needed for starting an infection and disease for each microbe. And each has their own port of entry, each have their own multiplication, attachment, and living site in the body, and port of exit. Mechanism of adhesion. So, for start of any disease, after finding suitable port of entry, the pathogen must attach. Okay? Must attach to the target tissue. So different microbes have different things for attachment. Attachment is very important. Some microbes have film Fimbria. Fimbria. Fimbria or ordinary pili. Ordinary pili. Fimbria or ordinary pili. So it is something look like sex pilus. Ordinary pili is look like sex pilus, but their numbers are more than sex pili, and they are very shorter than flagella, and according to the pili they can attach to the target tissue. So Fimbria, you can see bacteria by their fimbria can attach to the target tissue. Ordinary pili or fimbria are used for attachment. So as you know, flagella are used for movement, going from one place to another place. Flagella are used for movement, going from one place to another place. But sometimes some bacteria can use flagella for attachment as well. Attachment what? Attachment to the target tissue. Because for starting infection, it is very important to have a suitable adhesion. Some bacteria can use capsule or slimes or slime layer. Slime layer or glycocalyx or capsule. So according to this part, many bacteria can attach, can have adhesion to the target tissue, and then starting for penetration, starting for attacking, starting for using the tissue as the food. Okay, so capsule. There are some polymers around some bacteria. According to this polymeric substances, they can attach to the part. Cilia. So cilia are not present in bacteria. Mostly cilia are present in protozoa. Protozoa like Balantidium coli. Yes, like Balantidium coli. By help of cilia in Balantidium coli, sometimes, so you know, cilia are something like flagella, but they are very short and their numbers are too much. So all around Balantidium coli covered with cilia for movement. So the action of cilia is look like the action of flagella, but sometimes they can use cilia for attachment. For attachment. Okay, and here you can see it is virus. It is a virus. As you know, virus contain nucleic acid and capsid. And capsid. So all viruses need attachment by S. S means spike. S spike. All viruses have their own special spikes or peplomers. So by this spikes, they find their receptor on host cells. It is look like key, and receptor is look like lock. So by this key can open the lock and penetrate the tissue. As you see, for example, some viruses have the key of the liver, so they attack to the liver. Some viruses have the key for nervous system, so they attack to the nervous system. Okay? Some viruses have the key for intestinal cells, so they attack to the intestine. So we can understand why one virus cannot attack to whole cells of our body, because each virus have special keys, and these keys can only open special cells in our body. These keys are not matched to the locks of all the cells. For example, you have a key, you can open only one type of car. Yes? You cannot open the door of all the cars in the city, because your key is only matched to one car. And each virus keys can only match to a special type of cells. Okay, we call them spike. Spike. And in some worms or helminths, they have escolex. Escolex means head. And in their escolex, they have soakers. What do they have? They have head or escolex, and these heads or escolex equipped with some soakers, and by these soakers they can attach to the tissue, like, for example, Taenia saginata. So Taenia saginata is a worm. It is a helminth up to 10 meters and living in our intestine. And its life cycle depends on cow and human. So if we eat the meat of cow not well cooked, so we should cook properly the meat of the cow. If it is not well cooking, so the larva of this worm can come to our intestine and turn to the mature helminth with head and its head attached to our intestine by suckers, as well as by their hooks. So, for example, so Taenia saginata only have suckers, but Taenia solium has suckers plus hooks. Suckers plus hooks. What are suckers and hooks used for? Suckers and hooks in these types of worms are used for attachment. Okay, one of the student cannot hear me. Please, if the others can hear me, write me to understand. Okay, so Mr. Ali Abdullah Sadi informed us that there is sound. So, dear student Makarem, I think should checking the internet. Please check your internet okay. So the answer is please check it, because if one person can hear me, it is not the problem to Don, and this is not the problem for me. It is the problem for student with internet connectivity. So now in this page, we can go through a conclusion that all pathogens for starting infections need attached to the host tissue. Bacteria by fimbriae, by flagella, by capsule, protozoa by cilia, worms by scolex, by hooks, and so on. All microbes for starting infections need something, some tools, some devices to attach to the target tissue. So here you can see, this is a bacterium, for example. There are adhesion protein, and this is tissue cells. There are some receptors, like with carbohydrate. So for starting infection, this bacterium, by this fimbriae or ordinary pili, should attach to the tissue cell, and then infection can be started. Here you can see in bigger appearance. For example, this is epithelial cells, Escherichia coli, by its fimbriae attached to the receptor, and attachment is the first step for starting of the infection. Here you can see some bacteria which can invade and attach regarding their flagella. So normally, in most cases, flagella are used for motion, are used for motility, are used for going from some place to another place in liquid environment. But in some cases, flagella can used for attachment. Summary of the bacterial flagellum as an adhesion. So motility is the first role, but sometimes flagella have another role with the name of adhesion. Flagellum can mediate bacterial adhesion to the eukaryotic cells indirectly via motility. So number one, this bacterium wants to try for attachment or by binding directly. Number two, you can see flagella directly attach to the tissue, eukaryotic cells, to epithelial cells, either on apical. So it has apical or front part, and this is back part. So sometimes bacteria attach by the front part, but sometimes they attach by the behind part by use of their flagella to the target tissue. And you can see here there is attachment to the target tissue. Flagellar target receptors include mucus and mucins. So mucus and mucins in this part, you can see, can be used in epithelial cells for attachment. Here you can see flagella are used, and the head or front side are used for attachment. Different glycans on the cells or in mucus, glyconate, heparan, sulfate, proteoglycans, Lewis X, glycotype, GM1, acylo GM1, and extracellular matrix. All of these things can be used for helping attachment of bacteria to the target tissue. And this is the figure of Taenia solium, as I wrote a few minutes back to you. Taenia solium, it is a worm, something around 10 meters. It is adult. So I told you it has head, neck, and this is the body of the worm. This part is the head. They are 4 suckers around it. They are used for attachment, and this is hook, 2 rows of hook. So this worm by this suckers and hooks can attach to the intestine and defend itself not to take outside the body, because the worms wants to remain in the intestine by this attachment, this part for attachment. Okay, why do we talk about microbes, viruses, bacteria, protozoa? Because here we are talking regarding to the advanced microbiology. Okay, so microbiology is regarding to the different groups of microbes. It is not advanced bacteriology. It is advanced microbiology. Okay, the other part we want a few minutes to discuss is surviving Haas defenses. Who wants to survive? So there is host defenses attacking to the pathogen. So if one pathogen can survive host defense, it is very good to the pathogen. Yes, survive means not sensitive. When one microbe, when one pathogen can suffer, can survive, it means not to be killed easily by immune system. Initial response of host defenses comes from phagocytes. So the first army which in our body present for attack to the pathogens are, for example, neutrophils, macrophages. They attack to the bacterium and wants to phagocytose it. They want to eat and digest the pathogen. Wants to eat and digest the pathogen. So phagocytes are the first row. They are the first armies which attack to the bacteria. Ability to survive intracellular phagocytosis. So sometimes pathogens are phagocytosed, but not to be killed. They can continue their life. They can continue their life. And sometimes these bacteria have antiphagocytic factors. So they can continue their life by their defensive factors. As we have some defense against microbes, microbes have their special defenses also. They have their special defenses also. Antiphagocytic factors. So now we want to discuss what are the antiphagocytic factors. Okay. They are factors which they can prevent phagocytosis by hosts' phagocytic cells. Like what? The first one is bacterial capsule. Bacterial capsule. You know, some bacteria have great and big capsules around themselves. And sometimes 2, 3, 5, 10 bacteria come together with a very large capsule around them. Okay, so here you can see phagocyte come to the bacterium with pseudopoda, make around it. But if it has a large and big capsule, cannot send it inside, cannot absorb it, cannot phagocytosis. You can see the bacteria with big capsule can escape. For example, when we have a big pizza in front of us in the evening, okay, we cannot eat it because it is larger than our mouth. Yes, we should cut it to small parts. So when bacteria are very big, they are bigger than the mouth of the phagocyte and cannot be absorbed. So one way for escaping phagocyte is formation of big and large capsule. Bacterial capsule composed of chemicals not recognized as foreign. The other way, along with the size, sometimes bacteria can have some capsule similar the composition of some tissues and some parts of the body. So when phagocyte has responsibility to find those microbes which composition are different with the body. When bacteria cover themselves with some chemicals similar to the tissues of the body, so phagocyte going through misdiagnosis, not correct diagnosis, and think this part is a part of the body without get it going away. Slippery, difficult for phagocytes to engulf bacteria. So number one, phagocyte think it is one part of the body. The second part, as much as the size of the capsule is larger, phagocytosis is more difficult. And from one size more, it is no possibility of phagocytosis. It is number one. Number two, antiphagocytic chemicals. Some bacteria can excrete, can send some antiphagocytic chemicals to prevent fusion of lysosome and phagocytic vesicle. So when one bacterium is ingested, phagocytic vesicle should fuse with the lysosome. That with the presence of antiphagocytic chemicals, fusion of lysosome and phagocytic vesicle interrupted, prevented, not happened. Without lysosomes coming to this part, so this part is a suitable place for living, eating, growing, and multiplication. So not only not to be killed here, now here is a place for living and multiplication. Another thing, another antiphagocytic chemicals are leukocidins. Leukocidins directly destroy phagocytic white blood cells. So in this part, sometimes by some chemicals not allowing lysosome coming to the phagocytic vesicle, so this cell is alive, and inside this alive cell multiplication is continued. But sometimes This bacterium produce some substances and kill the phagocyte, can kill this army, yes, and escape from its attack. Enzymatic destruction. Enzymatic destruction. So after finding port of entry, after reaching to the target tissue, so now it is time for enzymatic destruction. Extracellular enzymes or exoenzymes are produced, are secreted from the pathogen. So extracellular enzymes or exoenzymes are secreted by the pathogen, dissolve structural chemicals in the body. So when bacterium finds the tissue, wants to penetrate it, wants to go in the depth of the tissue and use it. So excrete exoenzymes, and it helps pathogen maintain infection, invade, and body defenses. So when going to the depth of the tissue, going far from the immune system. Here you can see invasive bacteria rich epithelium surface. So it is epithelial cells, yes, and this is bacterium. Bacteria are present here. This bacteria wants to go in depth of the tissue. For example, they can produce extracellular enzyme. One enzyme is hyaluronidase. By producing this enzyme can produce a halo between epithelial cells and accordingly reach directly to the depth of the tissue. Then the other cells destroyed by collagenase. This collagen structure can destroyed by collagenase and invasion to the depth of the tissue is possible. You can see they can go to the depth of the tissue. Bacteria invade deeper tissue. Here, for example, coagulase and kinase. Bacteria produce coagulase. Here you can see coagulase. Okay. So by this enzyme can penetrate going to the blood, and sometimes there are some clots. There are some clots from red blood cells, and bacteria later produce kinase. By kinase, they break the clot and they can reach to the blood, dissolving clot and releasing bacteria to the blood. If bacteria are present in the blood for a short time, we call it is bacteremia. But if they can suffer immune system, they can go all over the parts of the body by blood. They can have multiplication. Then we call infection is septicemia. and septicemia is very dangerous. Yes. For example, if one enemy come to the country, first thinking about the roads, wants to get the roads. So blood vessels are the roads of the body. All food, all chemicals, or all red cells or immune cells, antibodies, all the things going and come back from blood veins. So if one bacterium can be in blood veins for a long time and can have multiplication, 100% can kill the person. So septicemia is very dangerous, infectious. So as I told you, after attachment, now it is the time for invasiveness, to going to the tissue, to the depth of the tissue, far from immune system, and use all the foods inside the tissue. One tool, one enzyme, you know, at the end of any enzyme we can see A's, yes, coagulase, streptokinase, hyaluronidase, and some another enzymes like this. Coagulase form a blood clot that protects them from phagocytosis. Sometimes bacteria wants to escape from phagocytosis of macrophages and white blood cells. Then they use coagulase. It is using for production blood clot. Sometimes they use streptokinase. Dissolve fibrin clots. It is in the opposite direction and allows dissemination of the bacteria. So opening the clot, production the clot, removing the clot. Hyaluronidase enhances pathogen penetration through the tissues. So you know one part of many cells is hyaluronic acid. So when bacteria produce hyaluronidase, can destroy hyaluronic acid and penetrate the tissue. Leukocidins disintegrate neutrophils and macrophages. So by leukocidins, bacteria can kill leukocytes like neutrophils and macrophages. Hemolysins dissolve red blood cells. So red blood cells contain many nutrients. Red blood cells are full of nutrients. So bacteria, for finding food, sometimes they use hemolysins to destroy the red blood cells and use their nutrients. Virulence can be enhanced in biofilms because immune system cannot reach bacterial cells. Okay, so till this part it is sufficient. Please next time tell me the last part of the page of 22. We continue. Next time I should start from this part. Virulence can be enhanced by biofilm. We are here. Okay, is it any question? Yes. According to the situation, if it is needed to escape from neutrophils, macrophages, making clot. If they are too much and they don't afraid from leukocytes, they want to come out and going many places, then it is the time for clot dissociation. Sometimes needed to produce clot, sometimes needed to dissociate clot. So it is according to the situation. Okay, thank you very much. Hope the best. So next time, please tell me last line of the page 22. Thank you very much. Hope the best. I'm glad to meet you again. And we will follow next week. Goodbye.